started it.
So we'll just wait a few minutes to let people join. Okay, so it's 12:02, so I'll just get started. I'd first like to acknowledge the traditional owners of the land on which we are meeting, and I pay my respects to the elders past and present, and the Aboriginal elders of other communities who may be here today. Good afternoon, everyone. Thank you for joining this month's webinar titled Smoke Pollution and the Immune System. I'm Sabrina Idris, a Centre for Safe Air Postdoctoral Research Fellow, and I'll be chairing today's webinar. Before we start, I'd just like to say that the Centre for Safe Air has released an open letter calling for a national clean air strategy with 14 like-minded organizations. You can read the letter and add your name on the open letter, and Bill will share the link in the chat shortly. Anyway, the immune system may hold the key to understanding the complex mechanism whereby air pollution harms the whole human body. Inhaling air pollutants such as pollen or fine particulate matter such as PM2.5 starts a cascade of immune system changes. Some parts of the immune system may become inflamed and stressed, while other parts are suppressed. To help understand these complex causal mechanisms and their health impacts, this webinar will feature presentations from two leading researchers, Professor Angela Haczku and Dr. Dwan Vilcins. Each speaker will present for up to 20 minutes, followed by a Q&A after both presentations have ended. So please drop your questions and comments in the Q&A section. Our first speaker is Angela Haczku, who is Professor of Medicine, former Associate Dean for Research and Director of the Lung Center at the UC Davis School of Medicine. She earned her MD from the University Medical School in Debrecen, Hungary, and her PhD in Immunology from Imperial College London. Following postdoctoral training at National Jewish Health in Denver, she served on the faculty at the University of Pennsylvania. Angela's research is focused on the immune mechanisms driving chronic airway inflammation in COPD and asthma. She is particularly interested in the effects of environmental exposures. She's published over 100 scientific papers, with her work cited nearly 7,000 times and awarded multiple funds by the NIH. She's an elected fellow and director at large on the board of the American Academy of Allergy, Asthma, and Immunology, and serves as chair of the Mechanisms of Asthma and Allergy Inflammation Interest section. She has contributed her expertise to numerous NIH and global advisory panels and serves on the editorial boards of leading journals in the field. At UC Davis, she holds the Chester Robbins Endowment for Pulmonary Research. Over to you, Angela. Would you like to share your screen?
Yes, absolutely. Thank you so much for the nice introduction, Sabrina. Sorry, that's the wrong. I don't know if
The screen was fine just now, Angela. Like we could see your slides. So all okay.
I see. Okay. All right. So
That's good.
good. What I wanted to talk to you today about was the focus of our recent investigations that's on these relatively newly discovered innate lymphoid cells. These lymphocytes are very interesting because they might play a profound regulatory role in mediating environmental exposures. And before I start, I wanted to acknowledge my laboratory. I don't know if you can see my cursor. My fellows, pre-doctoral and postdoctoral trainees, and also my collaborators from the pulmonary and critical care division who helped me with the clinical part of the study. To study airways diseases and pollution is very important to me because, as the American Lung Association says, when you can't breathe, nothing else matters. I put a few photographs here, some of them courtesy of my colleague Amir Zeki, that we captured in Sacramento during wildfires smoke events. And those events are not as rare as you imagine. Practically, we have them repeated almost every year. I'll show you a little bit more data about that. But I just want to introduce the role of the exposome, because the lung is exposed to the outside environment, which is a complex, multifactorial interplay of climate change related factors, behavioral stressor exposures, occupational, dietary, infectious, allergenic, and toxic air pollutant exposures. To measure the air pollution level, we usually use the EPA established air quality index. And here I depicted the two most commonly measured and investigated components of air pollution, the particulate matters and ozone. Particulate matters and ozone levels fluctuate wildly, and you may appreciate in this timeline that I depicted here between 2020 and 2023 that there have been some really huge events. In August 2020, we had the LNU Lightning Complex fire, which was one of the largest, if not the largest, in California's history. And then in 2021, the Caldor area fire, which resulted in the greatest exposure, toxic exposure levels. But this regularly comes back and almost every year is repeated. So when we are exposed to these toxic materials that we inhale, what happens is that the airway epithelium that protects our respiratory tract will become alarmed, and appropriately, the molecules that are produced in response to toxic, allergenic, or infectious exposures are called the alarmins. I would like to highlight three of those alarmins. There is a huge list of them, but I wanted to mention three of those because they have been relatively recently discovered. And what they do, actually, they serve as biological growth factors for those innate lymphoid cells that also live in our respiratory tract mucosal system. So you can see on the left side a photograph that's been pasted together during clean air and during wildfire smoke of the Bay Bridge that connects San Francisco to the mainland. And accordingly, we made some measures in serum released molecules, the eosinophil chemoattractant eotaxin 2, CCL24, the serum surfactant protein D, which is a lung injury related molecule released during lung injury, and serum D dimer, which is a general pro-inflammatory molecule. I hope you appreciate the difference between wildfire smoke and clean air, both in our serum circulating material and also in the cell types that we measured by flow cytometry that showed that these innate lymphoid cells that I mentioned you about become activated parallel to inhalation of wildfire smoke. We quickly were able to recruit a few participants in this study during a wildfire smoke event, and also re-recruit them 6 months later. Actually, we were only able to re-recruit 3 of them, but I hope you still appreciate the difference between the activation of these cells that increases dramatically during wildfire smoke. These are all healthy volunteers without any visible or pathological symptoms at the time. So the innate lymphoid cells that I'm very highly interested in derive from common hematopoietic innate lymphoid progenitors. And according to the dogma, they differentiate into 3 main subclasses, ILC1s, 2s, and 3s, depending on the transcription factor they harbor and the cytokines they release. ILC2s are classified as GATA3 positive cells that release the type 2 group 2 molecules IL-5, IL-13, and IL-9. Interestingly, when we tried to mimic these wildfire smoke exposures in a mouse model and we exposed them to one of the components, ozone, we found that there was a influx of neutrophilic granulocytes into the lung a few hours following exposure of the animals to ozone. In fact, you can see on the top right-hand side here, the time course of the neutrophil influx that denotes a transient inflammatory response. And this inflammatory response in the animals actually resulted in functional differences, which is a significant increase in methacholine responsiveness in measured as lung resistance. You can see in the bottom here that the number of innate lymphoid cells, especially group 2 innate lymphoid cells, also significantly increased when the animals received an inhalational exposure to ozone. So, wanting to see what are the molecules driving these functional changes, you probably all know that neutrophil influx in any organs, especially in the lung, is not a good thing. It causes inflammation, and as you saw, it caused inflammation and functional dysfunction of the airways. So we wanted to see what are the molecules involved and performed an Affymetrix chip on the genes that got activated in the lung tissue. Surprisingly, we found that a lot of these early events, just a few hours after after ozone exposure, IL-17A seems to be a focal cytokine. Now, going back to the slide when I showed you that group 2 innate lymphoid cells are not characterized originally by IL-17A expression, they are known to be produced by T lymphocytes and group 3 innate lymphoid cells. This activation of IL-17A gene actually leads to airway neutrophilia. You can see here that IL-17 release in the airways is very strongly positively correlated with all the other cytokines responsible for drawing neutrophils into the lung. And the IL-17 signaling pathways are activated as early as 2 hours after initiation of the toxic exposure. Interestingly, T lymphocytes that are shown here with the green histogram are not activated to produce any of the IL-17 in the lung. And as I mentioned to you, the ILC3s that are also classified as the IL-17A producers weren't activated either. So where does IL-17A come from? Surprisingly, we found that ozone-induced activation of the ILC2s resulted in abnormal, non-canonical cells that express not only IL-13 but also IL-17A. This expression was strongly associated with activation of the IL-17A driving transcription factor ROR gamma T. Interestingly, the alarmin that would drive activation of ILC2s, particularly IL-33, that is also the known major growth factor for the group 2 innate lymphoid cells, was accompanied by expression of IL-1 beta as well as IL-23 in the mouse lung of ozone-exposed animals. This is important because these cytokines, IL-1 beta and IL-23, are the main inducers of IL-17. When we reproduced this experiment in vitro in isolated group 2 innate lymphoid cells, we found that IL-13 expression was extremely highly upregulated when the cells were stimulated with IL-33, shown with the green circles. But interestingly, when we combined IL-1 beta and IL-23, they were able to drive ILC2s to produce IL-17A. And we were able to repeat this experiment in human cells as well. You can see on the left-hand side from a study that was performed by other investigators that IL-1 beta drives expression of Notch1, which is a receptor that can be found on the ILC2 surface. When we incubate these peripheral blood human ILC2s with a cocktail containing IL-23 and IL-1 beta, we will see a similar increase in IL-13, IL-17 dual producer ILC2s. And this effect can be also reproduced when we treat the cells with DLL1, which is the Notch1 receptor ligand. So both mouse and human cells can produce IL-17, but is this really important? To study this, we performed adoptive transfer experiment in which donor ILC2s were injected into recipients of animals that were deficient in RAP2 gamma chain. This deficiency results in lack of any lymphocytes, including the innate lymphoid cells in these animals. As you can see, the results, lack of lymphocytes and group 2 innate lymphoid cells causes a significant decrease in neutrophil influx in ozone-exposed animals. However, when we inject group 2 innate lymphoid cells back into these mice, we can replenish the neutrophil influx upon ozone exposure. You can see here that the number of ILC2s recovered from the airways positively correlated with the number of neutrophils in response to ozone exposure. Interestingly, however, when we also treat these animals with an anti-IL-17 neutralizing monoclonal antibody, this replacement effect is completely abolished. So group two innate lymphoid cells non-canonically can express IL-17, and this expression is biologically important, and it drives inflammation in response to environmental exposure. To look at the functional significance of these mechanisms in a higher species, we turned to our rhesus macaque center. We have a colony of about 5,000 macaques at UC Davis, and some of these animals, they live in safari conditions, and they are being followed from childhood. They spontaneously develop asthma. So we recruited some of these asthmatic macaques into our study and looked at peripheral blood cellular profile. Interestingly, we found IL-13, IL-17 dual expressor group 2 innate lymphoid cells in the circulation of these animals and stratified the animals according to high and low expressors. Interestingly, those macaques that express IL-17A at a high level in their ILC2s are also higher expressors of IL-22. They have greater number of ILC2s in their circulation, and most importantly, their lung function is much worse than those animals that do not have these profoundly abnormal cells in their circulation. To look at whether ozone exposure in the macaques would induce similar changes to the mice, we repeated the experiment and exposed these animals to ozone. You can see here that in the lung tissue, ozone induces a mixed neutrophilic and eosinophilic inflammation in the asthmatic macaques. And indeed, it was shown to be much more severe when the macaques also had these IL-17 positive group 2 innate lymphoid cells in their circulation. Interestingly, when the animals were treated with some antioxidant, the lung functional changes were alleviated, indicating the role of oxidative stress in this mechanism. Interestingly, in Davis, we show that we have a great level of air pollution. We did a study in which we expressed the cumulative PM2.5 exposure level over the map of California, and our COPD and asthma patient catchment area showed up very highly in this area. So we recruited some asthma patients and also looked for these IL-17 positive ILC2s and found that indeed some of the patients had these cells in their circulation. Interestingly, activation of these cells was strongly associated with myeloid pro-inflammatory dendritic cell presence, also presence of activated NK cells, CD4 and CD8 lymphocytes, and a strong correlation with the number of eosinophils in these severe asthma patients. Most importantly, the lung function in these patients was severely impaired compared to those patients who had none of these IL-17 positive ILC2s in their circulation. Similarly, recruitment of COPD patients from the same catchment area shows that COPD patients have a high level of mixed inflammation. Eosinophils don't normally characterize COPD, but in our population, it appears to be a predominant feature. Interestingly, such high level of eosinophilia was strongly associated with the number of IL-17 positive ILC2s in the sputum of these COPD patients, and also a worsened lung function in this subpopulation of COPD. So in summary, we showed that wildfire smoke exposure induces lung injury and activates ILC2s in healthy subjects. And ILC2s in a mouse model were necessary and sufficient to elicit airway neutrophilia as well as hyperresponsiveness in response to ozone, and activated the IL-17A pathway. Asthmatic rhesus macaques and human asthma patients showed the presence of circulating IL-17A positive ILC2s, and these cells in the sputum of COPD patients was a marker of severe mixed eosinophilic and neutrophilic airway inflammation and lung function impairment. So putting it all together, we propose that air pollutant exposure induces a non-canonical abnormal presence of IL-17 producing group 2 innate lymphoid cells, and these cells mediate and propagate a severe glucocorticoid treatment resistant neutrophilic and eosinophilic inflammation. So I would like to close with a quote from one of the activists, Greta Thunberg, who said, "I don't want your hope. I want you to panic and act like the house was on fire, because it is." And this photograph was taken just a couple of months ago in Los Angeles during the Los Angeles wildfires. Thank you for your attention, and looking forward to your questions.
Thank you so much, Angela, for sharing you and your team's incredible body of work. There are some questions in the chat, but we'll come back to them after all the presentations have ended. Our next speaker is Dr. Dwan Vilcins, who is the Group Leader of Environmental Epidemiology within the Children's Health and Environment Program at the University of Queensland. Dwan and her team explore the effect of environmental exposures on children's health, with a current focus on air pollution and plastic chemicals. She leads the NHMRC-funded AMBER study exploring the effect of bushfire smoke on health outcomes for several population groups. In addition to her PhD and Master of Public Health, Dwan has a background in nutrition. Over to you, Dwan.
Thank you, Sabrina. Oh, can you see slides? No, it doesn't look like you can.
Not yet.
My apologies. Okay, is that better?
Yes, that's perfect. Thank you, Dwan.
Okay, perfect. Thank you so much for having me here to share some information about the AMBER study today. AMBER is the Environmental Measures of Bushfire Smoke Exposure and Respiratory Health study, which is run out of the University of Queensland. And I'd just like to acknowledge our funders at this moment too. Sorry, my apologies. Okay, so the AMBER study uses a hazard reduction burn model as a proxy exposure of bushfire smoke. And while it's an imperfect model, there's several reasons why we think that it acts as probably the best proxy model that we do have at this time. Firstly, the planned nature of hazard reduction burns allows us to collect baseline data on participants before they're exposed, meaning that each person can act as their own control and reduce some of that background confounding we get when we're doing ecological or cross-sectional studies. It's also reliable during a funding or study period, which is obviously heavily desirable to funders. And it's also the most similar model of vegetation fires that we do have available to us, and also allows us to assess the penetration of smoke into households. A hazard reduction burns produce lower intensity fires and more smouldering emissions. And in the past, that's been held up as a reason of why it's not a great model. But in fact, as the evidence on the pollutant profiles emerge from hazard reduction burns, we see that these smouldering emissions often produce more pollutants, particularly fine particles, than the higher intensity flaming fronts. And wild and unplanned bushfires themselves also contain large amounts of smouldering emissions. So it is also a really important part of the fire to understand. In saying that, AMBER does apply its model to unplanned bushfire events that occur during the study period as well. So hopefully we can get some good data on both types. I'm not going to talk about everything with AMBER today. It's quite a broad study with can kind of be defined into three major study areas. The first is that we're studying what type of pollutants are present in smoke plumes and what pollutants remain in the environment within the charcoal and ash, which become a more chronic exposure source after the acute wildfire event has passed. Our second study is assessing what impact bushfires have on indoor air quality for nearby homes and whether short-term smoke exposure leads to measurable health effect. But the one that I'm here to talk about today is our work with the firefighters that are conducting the hazard reduction burns, where we're looking at their short-term health effects from being exposed and also looking at some preventative interventions. So the key tenet of EMBA is to really start to understand the causal and biological mechanisms that drive the clinical health effects of exposure to bushfire smoke. So we know from the epidemiological literature that being exposed to bushfire smoke is associated with a lot of health outcomes. But until we understand this biological pathway, we won't really understand what areas we can intervene. And so EMBA seeks to understand the internal dose that we're exposed to and biologically effective doses through biomarkers of exposure. And then also to understand how these internal doses lead to early biological effects and altered structure and function internally. And we use biomarkers of effect to understand that, such as oxidative stress and inflammation. And we also use a very sensitive measure of lung function called intrabathyoscillometry, which allows us to look at the effects in the very fine airways, not just the bigger airways. And so this allows us to pick up changes within the lung before overt clinical disease has arisen. And we look all the way down to clinical disease where we ask about symptomology. This is actually me performing the lung function test. It's quite easy. That's my daughter doing it on me. So of interest in this particular topic is our focus on oxidative stress and inflammation in particular. And I need to acknowledge Yu-Ya Chen, a very talented PhD student from the Queensland Alliance of Environmental Health Sciences, for this figure and for her work in analysing all of our urine samples. So, of course, the process of oxidative stress occurs when either internal or external sources of oxidants, such as reactive oxidant species, outweighs the body's inherent ability to prevent that type of damage, either through antioxidant enzymes or other types of antioxidant defenses. And that's where we get to cell damage. Within EMBA, Yu-Ya and her supervisor, Fisher Wang, have given us 4 different types of oxidative stress mechanisms. So we have the nucleic acid oxidation, referring to damage caused by DNA and RNA from reactive oxygen species. And these are really important for the immune system because elevated levels can lead to chronic inflammatory diseases. We have our lipid peroxidation, which occurs when free radicals attack lipids with double bonds, particularly the polyunsaturated fatty acids. And she's given us a few different ones in the panel here. We also have oxidation of sugars, where proteins and sugars bond to create advanced glycation end products. And then when they become oxidized, they can form quite nasty metabolites that can go on to lead to metabolic disorders and immune dysfunction. And lastly, and probably of particular interest to us as a respiratory focused group, is oxidation of proteins, where we have things like bromotyrosine and chlorotyrosine, which are produced by processes that involve neutrophils in the lung and the enzyme myeloperoxidase. So it's really nice that we have such an extensive panel with all of these different types of oxidative stress mechanisms, so we can kind of dig into which ones we think the bushfire smoke is most likely to induce. Just briefly to give you a sense of the methods, I thought I would present a day in the life of a firefighter enrolled in EMBA. So these are our firefighters in the Brisbane City Council area that we work with, and we turn up to a hazard reduction burn in the morning. Typically, we try to get there before the firefighters themselves arrive. So as they get there, we can have a little chat with them. If they're already enrolled in EMBA, we'll ask if they'd like to participate for this particular burn. Sometimes they're just too busy and they can't, but most of the times they'll say yes. And then we'll also recruit new participants. Before the shift, we collect a urine sample, we do a lung function test, and we ask for medication taken in the last 72 hours, just so we can see if there's anything that may interfere with our tests. After the shift, we turn back up. So this is at the end of a day of the hazard reduction burn, I've arrived and set up here before the firefighters have started to come off the fire field. And so what we do is that as soon as they come off, we grab them and we try and collect a urine sample, do the lung function test, check if they took any medications during the day. They have a very busy physical job, so often they're taking Nurofen and other anti-inflammatories throughout the day. And we also do some symptom screening. We're essentially using a modified ISAC questionnaire, which I know seems funny because that's developed for children, but because we're also enrolling households with adults and children nearby, having a modified ISAC allows us to kind of screen across the different population groups. And we find it holds up quite well with our adults as well as the children in our study. This is us doing lung function in the evening, and you can see that we've still actively got fires burning in the background. So it's always a bit exciting going out with the firefighters. We also get them to do a short online survey, collecting their relevant health info, and we return after 2 weeks of no exposure to any smoke to do all our sampling again to have a look if any changes have resolved by that time. I'm going to present some very, very early results at this stage. And I really do need to stress that these are preliminary results. Ember is in its first year, or it's just started its second year of a 3-year study. And so we analyse and keep an eye on the data as it's coming in, but at this stage, none of our results are finalised. So as I said, we're trying to understand the spectrum of exposure internally and how that leads to clinical disease. So when we look at our biomarkers of exposure, we can see that 1-hydroxynaphthalene has an increase in the acute period that's quite significant. With this particular biomarker, about 40% are below the limit of detection, so that is something we'll have to address. But we find much better results with 2-hydroxynaphthalene, which is also very significant in the acute period, and 100% of our firefighters had measurable 2-hydroxynaphthalene in the acute period. Actually, I think across all the time periods. We also have 2-hydroxyfluorine, which did a little bit better in terms of only 12.5% were below the limit of detection. And we did look at a fourth one, 2-hydroxypyrrole, but with greater than 74% of the cohort under the limit of detection, it wasn't worth going forward with at this stage. So we can definitely see that occupational exposure to bushfire smoke is inducing these measurable metabolites, so we can start to get a sense of what's going on internally with exposure. So next was to understand our biomarkers of effect. So with our nucleic acid oxidation, we can see a significant increase after acute exposure. So we saw a roughly 80% increase in 8-hydroxyguanosine and an 88% increase in 8-hydroxy-2-deoxyguanosine. And these were both quite significant. This is not surprising because I feel like these two are the most studied in both air pollution and bushfire smoke exposure. And then with our lipid oxidation, we found a significant increase in 8-isoprostanes, but not with our non-neol mercaptic acid. I'm sorry, I can't pronounce that one. So definitely just the focus going forward on this one lipid oxidation biomarker. With our sugar oxidation, so far we've only got one of them back from our laboratory, but we can see a slight increase there in carboxyethyl lysine. And with our protein oxidation, this is one of the ones we were kind of the most interested in because they can indicate inflammation in the lung. So, and we found some quite interesting results. So our 3-bromotyrazine actually reduced in the acute period by about 51%, and this was a statistically significant finding. I also just need to point out that this p-value is wrong and I meant to change it and I forgot. I'm sorry. So it was lower than that. It was statistically significant. And it is hard to tell with this axis because of the outliers, but it was actually 100% increase going from 0.38 nanograms per mil to 0.76. So these are conflicting results. Chlorotyrosine is definitely linked to neutrophilic inflammation in the lung. However, both of these come about from increased activity of myeloperoxidase. So we don't have a very simple answer as to why they're conflicting at this stage, but it's definitely something we'll focus on in the next 2 years. I know the focus today isn't lung function, but I just wanted to show how we're going across that spectrum all the way through. We have limited data because our team is really healthy. They didn't take a lot of breaths in the test, so we didn't get quite as many cycles as we would have liked. But the early results show we have some more negative reactance values in the acute period, and negative reactance indicates a loss of elasticity or increased stiffness within the lungs. So that's not something we want to see for our firefighters. And when it came to that respiratory symptom questionnaire, 71% reported some type of respiratory symptom post-shift. Most of them were mild, but some did report some wheezing and more concerning results as well. So it's a bit of a flash intro to EMBA because we really are still only in those early stages. We're funded to continue at this stage until the end of 2026. But it's really exciting because these early results do show that our exposure pathway has some merit. We're able to find results all the way across from the biomarkers of exposure into effect and those early clinical changes. And also that kind of focus that we have within a lot of the work that we do as oxidative stress mediating and driving a lot of these processes, and including inflammation and immune-driven processes. We continue to sample the firefighters for the next 2 years, and we already have recruited some more firefighters, so we have a few more people to add to the study for this year, which is exciting. And I'd like to acknowledge the Centre for Safe Air, who awarded us some seed funding to work on a co-design intervention study with the firefighters. Talking with them, they've made it very clear that they don't just want us to study their health effects, they actually want us to look for a range of potential health protective things that they can do. And it doesn't just all relate around personal protective equipment, which out in the field can be quite cumbersome and sometimes is not suitable. A lot of the available PPE that's out there. So we've got some work this year to do around that, which is really exciting as well. And so hopefully the next time I'm talking to you, I'm presenting the results of our more sophisticated analysis, and you know, we'll look at longitudinally what happens with these firefighters over the next 3 years, or next 2 years, or 3 years in total. I just want to give a shout out to my team because, of course, no study is done by one person. It's always a team effort. We have a really wonderful team, both at UQ, at the Queensland Alliance of Environmental Health Sciences, at Louisiana State University, and the Centre for Advanced Imaging. And if you would like to learn more about EMBA, or if you live in Southeast Queensland and you're interested in signing up as a participant, where we actually come out and sample your house if you're exposed to a hazard reduction burn or bushfire smoke, you can get more information at the QR code. Thank you.
Thank you so much, Dwan, for sharing your fascinating research. I'm very excited to see the end results. Now we'll open the floor for any questions that the attendees may have. So we'll start with Angela. So Angela, I saw that you answered some questions from the Q&A, but there was a question from John, and you've looked at asthma and COPD, and John was wondering about interleukin-17 and how it is a central cytokine in the pathophysiology of psoriasis. And they're seeing an increase in psoriasis and psoriasis arthritis. Do you think the increase in interleukin-17 from air pollution could be the cause of the increase in psoriasis?
Yes, definitely. I think environmental exposures from any sources, and if they come through the lung and increase IL-17 production, can affect other organs. So there could be a general a systemic inflammation, and that's what we were trying to also look at in checking the circulating lymphocytes and circulating levels. In fact, in a newer study, we looked at serum IL-17 expression during wildfire smoke, and we found that even though at a low level, but those were still increased. So yes, and I'm happy to very quickly answer the ones that I answered in writing as well. So just a question about IL-8. Indeed, IL-8 plays a role, of course, in neutrophil migration. IL-17 just amplifies the IL-8 effect. They work with each other. They are positively correlated. And the other question was about my terminology and what do I mean by non-canonical cells. So it has not been used before, but what I meant was to emphasize that this is a new finding. We actually just saw a new paper came out from a McMaster group where they show in asthma patients the importance of IL-17 producing group 2 innate lymphoid cells, but it has not been widely accepted before, and our findings went against the dogma. But I just wanted to show that they are important and they play a role in the severity of disease.
Thank you so much, Angela. And Dwan, Michael had some questions. He also just made a comment about how there's validated respiratory questionnaires for adults, like this ACRHS. So maybe that's something that you could potentially consider as a questionnaire for the firefighters. He had a question about what frequency that did you use in the FOD measurements. Is it X5, AX5 or higher?
Okay, I'm not a respiratory scientist, so I hope I have this correct, but we're looking at the 5 to 30 hertz and the 10 hertz. So we run both of them.
Yeah.
Also, thank you for the recommendation for the questionnaire. I'll definitely have a look into that, adding that in. I do like that we can compare against the adults, the children, and then the firefighters, but I think it would be really useful to have an adult-focused questionnaire as well. So thank you for that recommendation.
Yeah. Thanks, Dawn. And how many firefighters have been recruited so far? Do you have a target sample size?
So we have 18 that were in the data that I just presented, but we've recruited up to 25 now. If we got, up to 23. If we got 25 out of our current cohort at the Brisbane City Council, I'd be pretty pleased with that. But what we are doing is working with other councils now and the Rural Fire Service to try and just improve our pool of who's available. Brisbane City Council, they do have quite a large potential pool, but a lot of them are seconded in. And so, yeah, we're hoping to open it up to other fire authorities.
Yeah. Thank you so much, Donna. And Faye also had a question. So she's interested if what sort of concentrations of PM2.5 were the firefighters exposed to during their shifts? Do you do some AQ monitoring?
At this point, we don't. I've actually just been writing a new grant application, and one of the things that I want to add into that is personal exposure monitoring for the firefighters. So for the households we have an air quality sensor indoors and outdoors, simultaneously measuring what they're exposed to. But for the firefighters, they don't. It's definitely something I would love to add in.
Yeah, thanks, Dawn. And on that note, were the impacts that you observe in regards to the household, was it very similar to what the firefighters were experiencing, in terms of the lung function and the biomarkers?
Yes and no. It was obviously to a much smaller degree, but we have found with the 2-hydroxynaphthalene that we can definitely pick that up in the households as well. So that's turning out to be quite robust in our data so far. And there were a handful of biomarkers that we could pick up in the families, but not actually in the acute period, in the post period. Which was a follow-up anywhere between 5 to 10 days later, depending on when we got back to those households. And we did see some early indications of lung function change, but nothing quite as much as we see. Like in the firefighters, it's quite consistent with those reactance measures, but in the households, it's not. And we did definitely find that children had a stronger effect than the adults did, which was something as a children's respiratory group that we were really interested in finding. So, yeah.
Oh, that's interesting. Faye has a question for Angela. So Faye said that she's aware that you did some work on wildfire smoke and COVID vaccine response. Did you want to make a quick comment on what you found? Was the difference of clinical importance? For example, should we defer immunizations on smoky days?
Yes, thank you, Faye, for this question. Very quickly, actually, the paper came out in the Journal of Allergy and Clinical Immunology. You can look at it. We did find both at 1 month immunoglobulin levels and the 6-month immunoglobulin levels of those who received their vaccination at high air pollution during wildfire smoke events was significantly lower than when people were vaccinated during clean air. However, we don't know yet if it plays a role in terms of protection against COVID-19 or the flu. We are now working on setting up mouse models to study this and see if these wildfire smoke or environmental exposure, PM 2.5 or ozone exposure, would reduce protection as well. And if that is confirmed, then definitely I think it should be recommended that, especially in vulnerable populations like the elderly, children, or people who may have a different immune response to vaccination or a reduced functionality of the immune system, that they would receive their immunization during clean air and not at the height of wildfire smoke events.
Thank you, Angela. I think so there's a question from Arif. So I think this is for Dwan. Does the cohort group all were equipped with respirator or mask, and will this be considered in further studies?
Yeah, good question. They are all issued with fairly robust PPE as decided by the Brisbane City Council's Workplace Health and Safety Officer. What tends to happen in practice is that the implementation of that PPE differs quite a lot across individuals. And one of the interesting things of working with the firefighters is realizing that it really is quite a burden on them sometimes to wear this, PPE at its highest standard. And so for some people, they think it's really important and they implement it really well. And for others, they find that it's snagging on trees, it's very hot, it's making it harder for them to breathe while they're already doing a really difficult job. One thing they have asked us to do that we'll be doing with them this year is they were given a very, very fancy clean air respirator, and they would like us to test the effectiveness. And so we are enrolling them in EMBA in a randomized repeated study where we'll randomize them across the year into either wearing or not wearing this very fancy respirator versus their usual practice for that individual, and see if we'll just do the normal EMBA protocol and see if that makes any difference. But as part of this co-design study we'll be doing with them across the year as well is to really ask them and dig into what type of respiratory protection do they need and is appropriate for them out in the field. So maybe we can start to look at some more intervention studies with those types as well.
Thank you, Dwan. Let me see. Yeah. And so Irish also said thank you, Dwan. And Angela, I think this could be the last question. So do you have any plans on studying the impacts of the recent Los Angeles wildfires on the immune system?
Yes, I have some friends who work at UCLA but they are actually ICU physicians. So they study ARDS patients, and we are trying to set up studies. Obviously, those will not be acute events, so they are not like from doing studies, but post exposure. And we would like to look at some functional outcomes and also how the immune cells are affected in the long term and whether they convey any differences in terms of chronic disease outcomes. ARDS is a very severe disease, and it often ends up in pulmonary fibrosis. And so the question is whether concurrent exposure to wildfire smoke would affect the severity, incidence, and prevalence of development of pulmonary fibrosis in these patients. So there are a lot of questions, and also what are the cells, what are the molecules that participate. So yes, these studies all need to be done. I'm not really sure how the NIH will be interested in funding any of these studies anymore, but we will see. Thank you for the question.
Thank you. Looking forward to it. So as we conclude today's webinar, I want to extend my sincere appreciation to Angela and Dwan for their excellent presentations on this topic. We hope the attendees have found the information valuable and insightful. If you have any further questions or would like additional resources, please don't hesitate to reach out to the Center for Safe Air. We look forward to seeing you at our next webinar. Thank you so much, and goodbye, everyone. Thank you, Dwan and Angela.
Thank you.
Thank you so much. It was very interesting. I learned a lot.